Welcome to Cancer Lesson 3.1, How is our DNA organized? The goals of this lesson are to examine the different components of the genome and explore how the organization of the genome can dynamically regulate gene expression to impact cell behavior. At the end of the lesson, the students should be able to explain how the eukaryotic genome is organized, describe the function of introns and exons, and explain how repeating sequences in the genome, called transposons, can affect genome structure and dysregulate normal genes. We will achieve these goals by exploring the human genome through the UCSC Genome Browser. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. In eukaryotic cells, the DNA template, called the genome, is divided into chromosomes that are made up of genes and regions between the genes called intergenic regions. Genes themselves are made up of exons, which code for amino acids, and introns, which do not. When a gene is transcribed, the messenger RNA, or mRNA, contains only exons and so is much shorter than its gene. Genes can use exons in different combinations to make different variations of a protein, so one gene can make many proteins. Both the intergenic regions and the introns can regulate when and where a gene is transcribed because they contain regulatory sequences. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual, or lesson plan, provides a minute-by-minute explanation of the lesson structure. And the student workbook provides additional explanation for students. Be sure to print the lesson worksheet and the homework worksheet. You will also need access to a computer lab. Note that in this lesson, we assume students have basic knowledge of the central dogma of biology, which is that DNA codes for RNA, which codes for proteins. But to ensure they remember it, there is a short review at the beginning of the Do Now. The key points of the Do Now are that genes contain exons that code for amino acids and introns that don't. The introns are spliced out when a gene is transcribed into messenger RNA. Genes are separated by intergenic regions, which contain regulatory sequences. Intergenic regions also contain transposons, which are DNA sequences that can jump around the genome and regulate whether a gene is transcribed. We begin by discussing how genes only make up 2% of the genome. This means that if random mutations to DNA occur, there is only a small chance of these mutations affecting our genes. In eukaryotic cells, all the DNA in every cell's nucleus is physically organized into bundles called chromosomes. Chromosomes are made of DNA that either codes for proteins and DNA that does not code for proteins. Genes are parts of DNA that code for proteins. Between genes are intergenic regions. Genes themselves have their own organization. They themselves have areas that code for amino acids, or proteins, called exons, and areas that don't code for proteins, called introns. While genes are only a small proportion of the genome, actual exons are the smallest proportion. The intergenic region is the largest proportion. The intergenic region controls gene expression via regulatory sequences associated with genes, and also via transposons. Transposons can jump around the genome and land in different places. They are important because they can affect gene transcription. Take a minute to review the central dogma to make sure students know that genes and RNA are made of nucleic acids and proteins are made of amino acids. Next, we take a closer look at how proteins are made. First, the DNA that makes up a gene is transcribed. This means that RNA polymerase, a protein, reads the DNA and creates a new nucleic acid strand that is complementary to the original DNA strand. This new strand is called the pre-mRNA. This pre-mRNA, like the DNA, contains the introns and exons of the gene, but it is single-stranded. Pretty soon after the pre-mRNA is made, however, parts of the pre-mRNA begin to be cut out. These parts are called introns for intervening sequences. The introns get removed or spliced out of the pre-mRNA. Once all the introns have been cut, only the exons, which stand for expressed sequences, remain, and the resulting strand is called mRNA. Different versions of an mRNA can be made by using different combinations of exons. This strategy is called alternative splicing. Because of alternative splicing, the 25,000 or so genes in a human cell can make more than 90,000 proteins. Up until fairly recently, the introns that make up nearly 80% of genes were thought of as junk, but now we know that introns often contain important information about when and where to transcribe a gene. The intergenic DNA between the genes can also affect gene transcription. Intergenic regions can be made up of regulatory sequences associated with genes that can promote gene transcription. They can also be made up of transposons, or jumping DNA. Some transposons have their own promoters. Promoters are sequences of DNA upstream or in front of the start codon of a gene that regulates where a gene is turned on. We will deal more with promoters later in the unit. It shouldn't be surprising that transposons can wreak havoc if they actually jump into an exon. But exons are such a small proportion of the genome that this is not very likely. However, transposons can still cause problems even if they only land near a gene because they may contain promoters, and thereby, if they land in front of a gene, can turn that gene on. The activity for this lesson will require access to the internet. Students will split up into groups to explore one of 5 genes commonly associated with cancer. They will explore the human genome on the UCSC genome browser and answer questions on the worksheet related to gene organization, regulation, conservation among species, and presence of repetitive elements. The teacher's version of the worksheet has instructions on how to best focus students on topics relevant to the lesson. The key points of the wrap-up are that only exons are transcribed into mRNA. Different exons may be used in different combinations to make different variations of a protein. Transposons can jump around the genome. If they land in an exon, they will affect protein function directly. If they land near a gene, they can switch transcription on or off. In the wrap-up, we review the answers on the lesson worksheet. First, the retinoblastoma gene had the largest difference in length between the whole gene and the mRNA because its introns were the longest. The next question introduces us to the concept of the poly-A tails, which are strings of adenine nucleotides that are present at the end of all mRNA strands. The poly-A tail is necessary for keeping the mRNA stable as it migrates out of the nucleus and into the cytoplasm. The cyclin D1 gene has the largest difference in length because its poly-A tail is largest compared to the other genes we look at in this activity. BRCA1 and retinoblastoma genes have the largest number of exons, but BRCA1 has the largest number of protein variants because it splices different exons together in more different combinations. The telomerase gene did not have high levels of the transcription modifiers, suggesting that it was not being transcribed in the cells used for this analysis. As organisms evolve, their genomic sequences diverge. One way to know how closely related species are is to compare their genomes and see how similar they are. Humans and rhesus monkeys are more closely related than are lampreys, a kind of eel, to either of them. There is evolutionary pressure to conserve exon sequences because these code for proteins, and therefore if you alter exon sequence, you change protein structure and function. Changes to the genome are also more likely to accumulate in intron sequences because they comprise more than 80% of the genome. Humans and rhesus monkeys will have more similar exons and introns because they are more closely related, whereas humans and lampreys will only have similar exons because they diverged much longer ago and have had plenty of time to pick up neutral mutations in their exons as well as in their introns. Remind students that we're talking about mutations in germ cells here. Mutations in somatic cells will not be passed on. Finally, students should now be able to deduce the relevance of transposons to cancer. If a transposon acts as a promoter and lands near a proto-oncogene, it can switch the gene on. Conversely, if a transposon lands in the promoter of a tumor suppressor, it can switch the gene off. For homework, have students read a short article from The Scientist about RNA transposons and how they may be involved in cancer. One of the most common questions students ask is, why haven't we evolved to get rid of transposons? Most transposon DNA is not harmful, therefore it is not selected against. Actually, there are some advantages to having extra DNA, such as increasing diversity and acting as a sink for mutations. At the end of the lesson, collect the lesson worksheets to assess students' explanations of the functions of introns and exons and how transposons can dysregulate gene function. We will build on the concept of how the genome is organized in future lessons when we learn how DNA mutations actually turn normal cells into cancer cells. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.